Children, good morning. Children, in last session I have discussed with you various hierarchical order of classification such as species, genus, order, family, class, phylum and kingdom. So, from the species to kingdom level I have discussed with you. Today in this session I am going to discuss with you two new topics. So, one is nomenclature, another one is tools used in the classification, okay, or tools used in the taxonomy. So, children, hope you know or you are familiar with the nomenclature. You are familiar with the nomenclature. You learnt in your lower section such as 7th, 9th all, okay. So, in that you had a topic such as what nomenclature. What is mean by nomenclature? Why ma'am is it necessary to keep a name for an organism to use worldwide? All questions you can arise. Yes, it is correct. To use a common name worldwide for an organism is essential. Why? Because all over the world there are 6,000 different languages are used. So, if an organism, if we take the organism or called in 6,000 different languages in all worldwide. So, it is difficult to keep all the 6,000 names for each organism is very difficult for us, okay. So, that is what scientists, to avoid this difficulties, they plan to keep a scientific name for all organisms based on some criteria. So, okay, this is called what nomenclature. When we speak about the nomenclature of organism, it should have a two naming system. That is what two names it should contain. Say example for this human being. The scientific name of human being is Homo sapiens. So, the Homo is a genus name and a species is a specific epithet. Like that, each organism has two names by scientist, given by scientist. This is called what? Binomial nomenclature. So, there is a rules to write the nomenclature. Scientist divided many rules to give a scientific name for each organism. According to this rules, we can scientists follow the naming of each and every organisms, okay. So, let us enter into the topic, children. Nomenclature, in all probability these words must be new to you, but they all mean human in different foreign languages. There are presently more than 6,000 languages in the world, and an animal can be named is more than 6,000 ways. Unfortunately, it is impossible for anyone to have a good functioning knowledge of most languages, and hence there arises a need for a universally accepted scientific naming system for all organisms. So, the process of assigning scientific names to animals or taxonomic group is called nomenclature. 2-mark question. What is taxonomy or define taxonomy? The process of assigning scientific names to animals or taxonomic group is called nomenclature. For example, worldwide the scientific name Homo sapiens denotes human. Classification and grouping were done to facilitate a deeper understanding of the unique characteristic of each organism and its interrelationship among closely related species, okay. So, the scientific name for each organism, it's depend on the unique characters and interrelationship among closely related species for the organism. So, this classification plays a vital role, this naming system plays a vital role in arrangement of unknown species based on their similarities and dissimilarities. So, by keeping the scientific name which is used to play a vital role in keeping the animals various categories based on their similarities and dissimilarities. Numerous characters are considering to give a name for organisms. What are the numerous characters? Morphology, study of external characters, genetic information, that is genes present inside the nucleus of every cells is called what? Genetic information. Habitat, what is mean by habitat? The place where the organism are living is called what? Habitat. And feeding pattern, what type of feeding it will do, whether herbivore, carnivore or omnivore. Adaptations, the children, our environment is continuously challenged with various changes. This changes leads to changes in the lifestyle of organisms. According to that, the organisms do some modification in its body or develop some structure in its body, and this is called what? Adaptations. And adaptation leads to variation, variation develop into evolution. So all these categories, they are considered for keeping the name. Then one of the primary responsibility of systemic biology is the development of biological nomenclature and classification. What are the primary responsibility of systematic biology? One is biological nomenclature, another one is classification. Nomenclature is not an end to systematics and taxonomy, but it is necessary in organizing information about biodiversity. So the keeping nomenclature will not bring an end for the systematic or taxonomy, but it is an essential information about biodiversity. Nomenclature functions to provide names for all taxa at all level in the hierarchy of life. Naming of the organism is done based on the guidelines. Okay, so guidelines given by ICZN. What is mean by ICZN? International Code of Zoological Nomenclature. So based on this norms, we can give a scientist can give a name for organisms. The scientific name ensures that each organism has only one name, not more than that. Okay, so for example, human, he has only one scientific name, that is Homo sapiens. Not other names are there for human. Binomial nomenclature. Biologists follow universally accepted principles to provide scientific names to known organisms. Okay, so biologists follow universally accepted principle. The principle or we are going to see later, children. Next topic we are going to see, to provide a scientific names to the known organisms. Each name has two components. As I said, each scientific name has two components. One is a generic name, another one is a specific epithet. This system of naming the organism is called binomial nomenclature, which was popularized by Carolus Linnaeus and practiced by biologists all over the world. Okay, who popularized in keeping the binomial nomenclature was Carolus Linnaeus. His practice, his concept was followed by or practiced by the biologists all over the world. Example, the national bird Indian peafowl, the Pavo cristatus, the national animal tiger Panthera tigris, and the Tamil Nadu state bird is common emerald dove. It's called a Calcopus indica. So these are the scientific binomial nomenclature given to organisms. Next, trinomial nomenclature. Tri means what? Three. Bi means two. If animal has two names as their scientific name, it is called binomial nomenclature. If an organism has three names as their scientific name, then it is said to be trinomial nomenclature. This naming system was proposed by Huxley and Strickland. Drinomes means three names: generic name, species name and subspecies names are coming under the trinomial nomenclature. When members of any species which have large variations, then a trinomial system is used. Then on the basis of dissimilarities, this species gets classified into subspecies. It is the extension of binomial nomenclature system which has an addition of subspecies. All the three names are set in italics and only the generic name is capitalized. If handwritten, then it should be underlined separately. Example, Corvus splendens splendens, Indian house crow. Okay, what they are trying to see, you have to keep only one thing regarding this binomial nomenclature and trinomial nomenclature, children. For binomial nomenclature, the organism has only two names. One is generic name, another one is specific name, okay, or species epithetic. Okay, in trinomial nomenclature, the organism has three names. One is as usual generic name and species name, and the third one is what, subspecies name. Okay, this what you have to keep in your mind, children. This is since the organism has three names as scientific name, it is called a trinomial nomenclature. So this all the three names are in the italic. When we are using the scientific name, it should be in italic letter. Okay, and as the generic name alone started with capital letter, other two species name and subspecies name should be started with a small or lower cases, and if it are written in the hand, it should be underlined separately. So this what the way we have to write the nomenclature, whether it is binomial nomenclature or trinomial nomenclature. So example for trinomial nomenclature is Indian house crow. The scientific name of Indian house crow is Corvus. It is a generic name. Splendens and splendens are the species and subspecies name. Then tautonymy, the practice of 2-mark question. What is mean by tautonymy? The practice of naming the animals in which the generic name and species name are the same is called tautonymy. Example, Naja, Naja, the Indian cobra. Okay, what is mean by tautonymy? Both the generic name and species name are same. Then it is said to be what, tautonymy. Example for tautonymy, Naja, Naja. This Naja is first Naja is the genus name, second Naja is the species name. So it's a scientific name of Indian cobra, and this kind of naming system is called what, tautonymy. What may be the reason for the extinction of dinosaurs? Okay, children. Next, the rules of nomenclature. It is a 5-mark question. Tautonymy is the 2-mark question. Rules of nomenclature, it is a 2-mark question. The scientific name should be italicized in printed form, and if handwritten, it should be underlined separately. Already I said the scientific name should be italicized in printed form, and if handwritten, it should be underlined separately. That means genus name underlined separately, species name underlined separately, and subspecies name also underlined separately. And as I said, the generic name first alphabet should be in uppercase, that is in capital letter. Then the species, the specific name should be in the lower case, that is in the small letters. The scientific name of any two organism are not similar. Already I said the scientific name of any two organism are not similar. The name or abbreviated name of the scientist who first publishes the scientific name may be written after the species name along with the year of publication. For example, lion, this is a Felis Leo Lin 1758 or Felis Leo L 1758. So the name or abbreviated name of the scientist who first publishes the scientific name may be written after the species name. Okay, if any scientist publish the scientific name of an animal very first, and the abbreviation of the scientist will be written after the species name. For example, Linnaeus first publish the scientific name for lion. So how to write the scientific name for lion is Felis Leo. Felis is the generic name, Leo is the species name. After that specific name, the scientist, abbreviation of the scientist who first kept the scientific name for this animal, Linnaeus. So L, which year he kept the name, 1758, all has to mention. And if the species name is framed after any person's name, the name of the species shall end with I or II or AE. So if the species name may be any scientist name. And how to write the species name? The species name should be end with either single I or double I or AE. Okay, for example, a new species of ground dwelling lizard, okay, Cyrtodactylus, has been discovered and named after the scientist Warren Gray. So that scientific name is what? Gyrtodactylus Warren Gray. That is, the Gray is end with what? Double I, not as single I. Next one is concept of species. What is species? Species is the basic unit of classification. Already we discussed in our last session. The term species was coined by John Ray, 1-mark question, in which in his book, what? Historia Generalis Plantarum. It consists of 3 volumes in 1693 itself. Describe the species as a group of morphologically similar organisms arising from a common ancestor. Carolus Linnaeus in his book Systema Naturae considered a species as the basic unit of classification. So species can be defined as a group of organisms that have similar morphology and physiology and can interbreed to produce fertile offspring. In 1859, Charles Darwin in his book Origin of Species explained the evolutionary connection of species by the process of natural selection. Okay, there are different concepts given by different scientists for the species. Okay, so John Ray only first coined the term species, and in his book Historia Generalis Plantarum. In this book, he described what is species. Species as a group of morphologically similar organisms arising from a common ancestor was the concept of species given by John Ray. Next followed him Carolus Linnaeus. He mentioned about the species in his book Systema Naturae, considered the species as the basic unit of classification. Species can be defined as a group of organisms that have similar morphology and physiology, can interbreed to produce a fertile offspring. And in 1859, Charles Darwin in his book Origin of Species, he gave a definition for species is evolutionary connection of species by the process of natural selection. By a natural selection, the evolutionary connection of species as given. So these are the various concepts of species given by various scientists in various period of life. Next, so tools for study of taxonomy. So children, so far I have discussed about the nomenclature of organisms. Okay, now I am going to discuss with the tools for study of taxonomy. Okay, what are the tools we are using to study of taxonomy? Okay, see children, tools and taxonomical aids may be different for the study of plants and animals. Okay, so we are using different tools to study plants and animals separately. For the study of plants, we are using herbarium and botanical garden used as a tool. Okay, when we are going for herbarium, you can see different types of plants in it. Okay, same way when you go for botanical garden, there you can find different types of plants in it. So that is the tool to study the what plants. So herbarium and botanical garden is the tools for study of plant taxonomy. And what about animals? How to study the animals? See, classical in the case of animal studies, the classical tools are museum, taxonomical keys, zoological park and marine park. These are considered as a classical tools which are used to study the animals. Arinyar Anna Zoological Park, also known as Vandalur Zoo, is in the southwestern part of Chennai. Tamil Nadu, spread over an area of 1500 acres, is one of the largest zoological park in India. Which is the largest zoological park in India? Arinyar Anna Zoological Park, where it is located in the west part of Chennai, Tamil Nadu. Its area is 1500 acres. The zoo houses 2553 species of both flora and fauna are found. In the zoos, there are 2533 species of both flora and fauna are present. Okay. The important component of the taxonomical tools. What are the important components of the taxonomical tools? Are field visit. When we go for a field visit, we can see many animals and we will identify the animals and we can take the list of the animals. Survey identification, classification and preservation and documentation. So field visit, and we will do the survey. In the field we are doing the survey of the animals and plants, and we will classify the animals based on its similarities and dissimilarities. And after classifying the animals, we have to preserve the animals and document about the animals, or the basic important component of taxonomical tools. Many tools are being used for taxonomical studies. Amongst them, some of the important tools are discussed below. So to study about the animals, many taxonomical tools are used. Some of the important tools are discussed below. Classical taxonomical tools. First one is taxonomical keys. Keys are based on the comparative analysis of the similarities and dissimilarities of organism. What is mean by taxonomical tools? It is nothing but based on the comparative analysis of the similarities and dissimilarities. They are find out the organism. There are separate keys for different taxonomic categories. There are separate keys for different taxonomic categories. See, then what are the different taxonomic categories? Species, genus, family, order, class, phylum. These are the different taxonomic categories. Okay, so each categories there are different keys are there. This is called what? Taxonomical keys. Next, museum. You know, children, in a museum there is a collections of preserved plants and animals are there, isn't it? So from that we can see many types of animals. So by then we can classify the animals and we can know the animals of different types. Biological museums have collection of preserved plants and animals for study and ready reference. Specimens of both extinct and living organisms can be studied. Next is zoological parks. These are the places where wild animals are kept in protected environments under human care. It enables us to study their food habitats and behavior. So, you know what is mean by zoological place? It is a place or vast areas in which animals and plants are protected in a natural environment. This is called zoological park. Okay. So, the next one is marine park. Marine organisms are maintained in protected environment. So, in marine park like zoological park, in a marine or ocean or seas, particular areas are announced as a protected areas. In that protected areas, the species of endangered organism can be preserved and maintained. Next one is printed taxonomical tools. Consist of identification of cards, descriptions, field guides and manuals. Okay, what is mean by printed taxonomical tools? About an animal, the photograph is printed in a card and about it a description also there and field guides and manuals also there. This is called what? Printed taxonomical tools. Okay children, what we discussed so far are called, that is taxonomical tools. Zoology museum, zoological park, marine park, printed taxonomical tools. These are coming under classical tools for taxonomy. Now we are going to see a molecular taxonomical tools. Technological advancement has helped to evolve molecular taxonomical tools from classical tools to molecular tools. The accuracy and authenticity is more significant in the molecular tools. Underline it children, it is very very important. In which in which taxonomical tools the result will be more accuracy and authenticity is what? Molecular taxonomical tools. The following methods are being used for taxonomical classification. Children, just you have to find memorized only the various taxonomical tools only. Okay, don't what it is, don't get confusion about the various method. Okay, because each one is very difficult. So to understand that also very difficult. Since we are having only this online classes, I couldn't explain without board also. So just you have to by heart the names. Okay, what are the various molecular technique which are involved in taxonomic molecular tools of taxonomy. Okay, so molecular technique and approaches such as what? DNA barcoding. It is a method to identify the organism. What is DNA barcoding? It is a short genetic marker in an organism's DNA to identify it as belonging to a particular species. Then DNA hybridization measures the degree of genetic similarities between pools of DNA sequences. Next, DNA fingerprinting, by which we can identify an individual from a sample of DNA by looking at unique pattern on DNA. Restriction, fragment length, polymorphism analysis, and in that is difference in homogeneous DNA sequence that can be detected by the presence of fragments of different length. Okay, these all you leave it children, just you by heart the various names alone. Next one is polymerase chain reaction sequencing are used as a taxonomical tool. See children, what are the things you have to learn? This is a homework also. Molecular technique using DNA barcoding, DNA hybridization, and the third one is DNA fingerprinting. Fourth one is restriction fragment length polymorphism. And fifth one is polymerase chain reaction sequencing. So these are the 5 tools used in the molecular level of taxonomic classification. Okay, so next one is automated species identification tools. Okay, how to identify the species automated? Okay, it consists of cyber tools. For example, what are the cyber tools? Daisy, Alice, Abyss, Spider, drawing, etc. are the various cyber tools used in the identification, species identification tool. What is mean by LS? Automated leaf copper identification system. We can identify leaf copper in that. Daisy, digital automated identification system. Abyss, automated bee identification system. Spider, species identified automatically. Spider, wasp, bee wing character are identified by this technique. Drawing, honeybee wing identification. So these are the various automated species identification tools. This is a 5-mark question. New taxonomical tools. This is based on electron microscopy images to study the molecular structure of cell organelles. So what is new taxonomical tools? It is a electron microscopic structure by which images through which we can study about the molecular structure of cell organelles such as mitochondria, ribosome, lysosome, endoplasmic reticulum, like this. Then ethology of taxonomical tools. Based on the behavior of the organisms, it can be classified. For example, sound of birds, bioluminescence, etc. Based on the sound of birds, we can classify the birds. Same way, bioluminescence also, by the character of bioluminescence also we can classify the animals. Then e-taxonomic resources. Inotaxa is an electronic resource for digital images and description about the species, which was developed by Natural History Museum, London. Then Inotaxa means what? Integrated Open Taxonomic Access is the expansion of what? Inotaxa. Okay, children. That's all. This topic over, and also this lesson also got over. And the homework for you is what? Define binomial nomenclature. Let's this one, it's a book where give a various concept of species, one question. And what are the rules to follow the nomenclature? This one, 2 questions. And write note on molecular taxonomical tools. These are the 3 questions. Give the basic rules used in nomenclature, first question. Second question, give the various concept of species. And third one is write note on molecular taxonomical tools. These are the 3 homework for you children. Complete it and send it to me. If there is any clarification, you can text me personally. Okay children, take care, bye.